Hello and welcome to a certified group webinar on what's hot in the beverage industry, a look at current trends and regulations. My name is Jessica Lloyd, Marketing Communications Coordinator with EAS Consulting Group, and please join me in welcoming today's presenters, Omar Oyarzabal and Alex Brandt. Dr. Omar Oyarzabal is a Senior Consultant for Food Services at EAS Consulting Group. Omar works with clients to develop food safety protocols per FSMA and HACCP regulations and facilitates customized client training programs. He holds many food safety certificates through AFDO, including Processing Authority, Seafood HACCP Lead Instructor, Sanitation Control Procedures, Foreign Supplier Verification Program Lead Instructor, and a Sprout Safety Alliance Lead Instructor. Dr. Alex Brandt serves as the Chief Science Officer within Certified Group. In his role as CSO, Alex combines his knowledge of food processing operations with his expertise in microbiology and molecular biology to provide technical solutions for Certified Group customers. This includes overseeing the Lab Plus scientific team who perform customized research studies and the technical services team that provide technical guidance to customers on method validations, regulatory matters, and contamination troubleshooting. As a reminder, our team will address questions following the presentation. Please submit questions using the Zoom Q&A feature. Thank you again for joining us, and I'll now hand it over to Omar.
Thank you very much, Jessica. Can you hear me well?
Yes.
Yes, thank you. Hello, everyone. The camera is over there. I have a little bit of a different setup around here, but let's go ahead and start with this webinar. I always try to present some of the acronyms that we use when it comes to regulatory compliance. As you can imagine, the process of just making regulation in this country is very complex, and there are so many different agencies. Regarding agencies, I will mainly focus on the Food and Drug Administration, although I may mention a little bit the Environmental Protection Agency when it comes to some of the drinking water, okay? There are some specific rules, laws that come from the Federal Food, Drug, and Cosmetic Act, and most of the regulation, pretty much all the regulation within the CFR, the Code of Federal Regulations, are intended to help comply with those regulations. So the topics I will cover are some definition, classification of this kind of complex family called beverages. We will talk a little bit about the fact that when companies are crafting beverages, they have to consider incorporating food safety at the very beginning. It's pretty much always better to do it earlier than later. We will talk a little bit briefly about acidified food and FDA, although that hasn't changed too much at all. And then we will talk a little bit about the trends. As we go, we can talk a little bit about FDA, inspection outcome, but I'm not going to focus too much on that. I prefer dealing with the trends. That is something that we see more. So let's jump right into that. What are beverages? Well, these are products that are in the liquid form presented as a conventional food under the Federal Food, Drug, and Cosmetic Act. So remember, beverages are considered conventional foods and are covered by those regulations. There are many different names, right? You can use beverage, some people use drink, water, sodas. We will see that some of them even have, special identification and a standard of identity when it comes to how it should be made. The labels also allow you to contain certain type of statement that are what we call generic statement, like refresh or hydrate, okay? The recommendation and directions for use and drink of these products are really meeting the definition of conventional food, like a source of fluid, like bottled water, or provide nutritional values, like in the case of milk. And they also use typical beverage type of criteria when it comes to the marketing. You can market it based on taste, the word refreshment, a thirst quenching. Okay. And of course, there are many, many different ways of classifying these beverages. One of them that I'm not going to deal too much with is the alcoholic beverages, right? That are under a specific type of regulation, the TTB, but also within the current good manufacturing practice by FDA for those that manufacture alcoholic beverages, like wine, spirit, hard seltzers. Potable water is more under the EPA regulation, what you would call drinkable water or drink water or water for drinking. That would be the tap water. They are really under EPA, who has set up the standards for that type of water. And of course, the state incorporate those regulations. We can have fermented liquid product that can be food or dietary supplements. We can have those are not really within, we will see that those are not, once you move to dietary supplement, that's not really a beverage. And we will talk about that a little bit. I will clarify that. Liquid probiotics, which can be also a dietary supplement or even a drug. And of course, if not a conventional food, okay? These are general way of classification, but with the caveat that probably the term beverage doesn't apply to them. Energy drinks, like functional waters, fruit juices, which follow under the juice HACCP, and non-alcoholic beverages. Basically, from all the new version that we have, including some of these hard seltzers or, hard teas, we go all the way to non-alcoholic beverage. And I'm going to mainly talk about some of these products over there in that corner. I'm not going to deal too much with the rest, okay? So we're talking about, in general, bottled water, carbonated products, coffees or teas, milk-based. And you will see that the vast majority of the beverages that we find that are within the beverage category is really, they use water as a main ingredient, right? Sometimes the water may be the only ingredient, like in the case of bottled water, that has a specific standard of identity. You can add other ingredients, and I use the term allow because we will see later that you cannot use an unapproved ingredient in some of these in the product because you will be violating some regulations, right? When it comes to ingredients, there are many different ones: liquid, solid, carbon dioxide, which is very soluble in water, is considered an ingredient, but it's part of the making of the water. Speaking of that, probably all of us remember the basic of how to make carbonated water. And I just took this little video from the internet because it shows a very simple process where you just have water and you add carbon dioxide, okay? The famous, the traditional term salt, right? But we can go all the way now to much more complex non-alcoholic beverages that tend to have similarities to alcoholic beverages, but without the alcohol. So over the last 2 or 3 decades, we have been changing a lot all of these different type of product. And even after COVID, we are changing also what people are requesting in relation to probably products that are non-alcoholic versus alcoholic products. I prepare a general diagram of a beverage manufacturing company just to show a little bit the basic, but at the same time, highlight the complexity of that. We mentioned that incoming water is going to be the main ingredient, of course. So incoming water is very important. That water may be treated if it is a well or groundwater, maybe even be treated if it is just coming from a municipality, right? Then we will mix that main ingredient with other ingredients that are in less concentration in the general formulation. Can be solid ingredient, liquid ingredient, right? And after mixing, we're going to move to the rest, but even the water can go through a new treatment before mixing, like UV light, filters. It all depends on the quality of water that the company would like to use, okay? As you can imagine, the better quality you have at the very beginning, the safer the water it is, the less amount of processing you have to do later, right? Very simple. Then you may add carbonation. Some people may use pasteurization, a thermal processing system before it goes to carbonation. All these thermal treatments are going to be under atmospheric conditions, so we can call them pasteurization system, right? Then they will be packed and they will be shipped. But if you don't add, a thermal treatment there, you may want to add it later, maybe in the way of a tunnel pasteurizer. As you can see, this is a very simplified general diagram, but the reality is that these plants are quite different. And once they are set up for one or two type of beverages, it's kind of difficult to add others that may require a special thermal treatment or a special type of mixing, okay, without altering all this process. When it comes to pasteurization, we do quite a bit of calculation of the temperature needed for products to be considered safe. There are many different types of pasteurizer, as you can imagine. We can have continuous one. We can have tunnel pasteurizing that can be relatively simple up to very complex pieces of equipment. You can go all the way to an aseptic processing. We have, dealt with several, acidified food beverages that are really packed in an aseptic system, okay? That aseptic system is, as you can imagine, a system that provides from an aseptic environment for the container and the product, and you fill the containers with the aseptic product in an aseptic, filler, an aseptic environment. Of course, these are not they're not inexpensive pieces of equipment or simple to access, right? So these are mainly for large volume of production. So keep in mind that we always encourage for you to develop and craft your beverages, but incorporate, the safety or talk about safety considerations as early as possible. If we go back to this diagram, you would see that there are tendencies, like people say, let's try to use and replace ingredients. Are you replacing ingredients with an ingredient that is as safe as the previous one? What if I remove the carbonation? What if I try to remove the temperature in a batch pasteurizer or in a tunnel pasteurizer? What is the impact of that in the safety? So those are very valid questions, and there is not a single answer. We cannot really address all of them right here in this webinar, but just keep in mind that just removing a processing step or changing some of the ingredients may have an impact on the safety of the finished product. There is always this question about acidified food and where they fit in there. A beverage can be an acidified food. There is no doubt about it. But it's in certain conditions, for instance, when it's non-refrigerating, non-carbonated. Remember, the definition of an acidified food is that you have ingredients that are low acid food that have to be acidified, and you want a product that will be shelf stable, okay? You don't want to refrigerate, for instance. So if you remove carbonation, refrigeration in some of the common beverages, they may fall within what would be considered an acidified food. Please consult with the processing authority when dealing with these changes. And we're going to review a little bit more this part. I'm gonna ask some questions as we go in the next 15 minutes that I have left. Question that we receive when people register. Like one question was, does lemonade need to be categorized? No, it depends. If the product is refrigerated, of course, it's exempt from the acidified food regulations. Another question we got that is an interesting one, I haven't seen it for several years, is back in 2010, FDA was drafting a guidance for the industry about acidified food. We have been working in foods for many years, so we have some copies, but now you cannot find it. So the question is, are there other similar publications? No. The answer is no, and the reason is acidified food are so complex in the different categories that covers, because it covers not only beverages, but a lot of other, conventional food, medical foods. Actually, dietary supplements may become acidified foods. So that's important to remember that, no, there is no specific guidance. Keep in mind also that when it comes to beverages, there may be some cases of standard of identities. Like, for instance, if I want to can a fruit juice, a canned fruit juice or a bottled water, they have a specific standard of identities that are very well described in the Code of Federal Regulation. Like, for instance, 146, 114 is for lemon juice, and 165, 110 is for bottled water, okay? So when it comes to trends, the first one that I want to deal with is the use or non-traditional ingredients or new ingredients that have been used in the last few years, or the ingredient that we will see coming into this product in the future. There is a shift a little bit about product with less sugar, different type of flavors, non-traditional ingredients like gingers, like now are in a liquid form, or flavors, right? So it's important to understand that the history of use of the ingredient is very important when evaluating the safety of the finished product. And if you change that history of use and use it in a different product, you may want to reassess and see if how we process that, because the actual processing of the ginger, I just put this as an example, may create a potential for hazard that we didn't have before, right? So make sure that you understand the safety of these ingredients. Make sure that you have the right suppliers, a good communication with the suppliers. Make sure that there is an evaluation of the safety to make sure that if you are using different ingredients or changing ingredients, you are still making a safe product. Again, keep in mind, water is the most important component, and we talked about allowed ingredients, and that's very important to remember, because if you add an ingredient that is not approved for the use that you are trying to make, you are violating some regulations, right? Some laws. Again, all beverages are treated as conventional food, and the ingredients really can be a grass substance, a food additives, prior sanctioned substances that are substances from previous to 1958. So they all have to be within some of these categories, right? Now we are facing new ingredients, or all ingredients in different concentrations. For instance, caffeine. Caffeine has been used for many, many, many years, but is that as the right amount? Is that a high amount that can be impact the safety? So keep that in mind. There are, because of the different type of changes going over the last few years, we have access to specific ingredients that we haven't used before. And thanks to the Farm Bill from 2018, now hemp, a cannabis plant with less than 0.3% of THC, can be added to ingredients, okay? So make sure you understand the use of the ingredients and the safety, okay? So we have a question. A beverage formulation containing herbal ingredients fall under conventional beverage or supplement drinks? Well, I think they were talking about dietary supplement drinks, right? So you cannot use the word beverage for dietary supplements. Beverages are not dietary supplements, okay? You may have novel ingredients that you may want to add in the labels, but please understand that you're still using them, all of these ingredients. You're selling the food with the labels of a conventional food. Once you start making functional claims with the product, that product is a dietary supplement. FDA will not consider that a beverage. It can be a liquid. It can be a liquid product, but it's not a beverage. Remember, beverages are conventional food, and you cannot market them as dietary supplement. If you want to claim and to have, you know, some specific functional claims on the labels, that product will be covered as a dietary supplement, okay? There are some recalls from time to time. We will see that in general, you know, beverages are quite safe, but sometimes there are things like this case that happened in Canada early this year, where they were adding a product that is considered a drug, okay? You cannot add a drug to food, right? Another trend that we see that is really the engine of the new crafting of many different beverages, that is the use of clean labels with less chemicals in the information, you know, in the formulation, I'm sorry. So there is a lot of words about, you know, healthier products. More people want foods with less ingredients in general, okay? In general, my take, and this is a personal take, clean for the public means chemicals. It doesn't mean necessarily less biological contaminants, because they are just focused on what is disclosed on the ingredient list, which are sometimes chemicals like preservatives, right? So just make sure that you review the labels. You have somebody with the expertise to review the labels and the potential claim that you're going to make, because again, there is a limited amount of claim that you can make with a food before it moves to being considered, for instance, a dietary supplement, okay? Another trend is less processing, okay? There is this concept that processing is bad, all processed food are bad, and sometimes people confuse what processing means, right? It's the same trend that we have had for food. Just keep in mind that there are some manufacturing steps that are critical for the safety, like a thermal treatment, okay? And if you remove that, you may impact in a negative way the safety of the product. So make sure that you understand what are the changes that you want to make, what are the tests that you're going to do to impact safety. And that doesn't go only just in general for formulation, goes for everything else that you change in processing. I got this little picture that I love because it shows how complex the overall manufacturing system of a beverage can be. Okay, so keep in mind that you need to keep, you know, an eye on that. The last trend that I have is a trend related to how to select a co-manufacturer for partnership. Sometimes we get those questions that are kind of beyond what we do in regulatory compliance, but understand that there are a lot of co-packers out in this world making a lot of beverages. A lot of the beverages that we consume are made by co-packers, right? So it's important to understand first the classification of the product that you want to do, how it's going to be, what are the regulations covering the product, to make sure that you are in the right positioning. So it's not a bad idea to have a processing authority to review the product process and say, this is what you need for safety. Then you need to really see if your potential partner, this future co-packer of yours, will have the appropriate equipment to manufacture a safe product, okay? Sometimes we have seen cases where, they are ready to move and start manufacturing, and then when we do the assessment, say probably you don't have enough for the manufacturing of a safe product. And it's not a problem with who developed the formulation, it's more the processing, okay? Or all of a sudden they have to change the processing to adjust to what the equipment they have in the co-packing facility. And it goes beyond just the equipment. Do they have the expertise? Do they have the programs? Sometimes to put together programs for safety, just for regulatory compliance, may require expertise and training that a co-packer may not have. So keep that in mind. So in general, as I mentioned before, beverages have a good history of safety. And in most of the cases that may be some cases of recall, these are not class 1 recalls. They are usually class 2 or even class 3, meaning that the hazard there are not a very significant hazard, okay? We have some examples. A couple of days ago, I went through some of the recall and market withdrawal and safety alerts from FDA. I found one, where you can find in here that it was really a case of a non-viral hepatitis. That's a particular, very unique case because it's a biological hazard, but non-viral hepatitis probably are coming from contact with a human that was the main vector introducing it to the beverage. Here we have an issue of some undeclared ingredients, like allergen that are considered a chemical, hazards, okay? So the final question I have in the last couple of minutes is, what impacts does the beverage industry anticipate from this new reorganization of FDA, where they're talking about the Office of Regulatory Compliance and CIFSAN? Very interesting one. Actually, very interesting, but there is no changes in inspection. I don't think there's going to be any changes. Many cases of reorganization of agencies do not impact inspection directly. They may impact how the organization and inspections are, but it's very difficult to change an inspection system that has been in place for decades. So that's all I have. I'm going to stop sharing. So Alex, you can take it from here. Thank you.
Omar set it up very well. I think here to talk about, the new trends that we have and see in beverages and beverage manufacturing. Really what I'm going to do is talk about microbiological aspects and some of the new trends that we're seeing on the microbiological front, which, I'm going to have a little bit of a bias, as I mentioned, to some particular categories like juices, for example. Just because, there's a lot more microbiological problems with those types of beverages, compared to others. Some may only have a spoilage issue. Some may actually have a pathogen challenge that they actually have to deal with. So I'm going to kind of frame it up in that regard. So I may have a little bit of a bias to certain categories and just kind of warning everybody on that. So first of all, kind of what I'll cover, pathogens and spoilage organisms that are concerned in the beverage processing industry. We'll talk about some of the detection technologies, some of the new ones that have been developed for those pathogens and the spoilage organisms in beverages. Then we're going to talk about control measures for mitigating those problematic microbes in beverages. So there are some, traditional control measures, and then we'll also talk about some of the newer processing technologies. And then how do we validate those control measures? So we can implement a new novel process for doing pathogen reduction in a beverage, but how do we actually go about validating that that technology is producing the intended effect? And I'll kind of talk about that here at the end as well. So where do microorganisms come from that we have in beverages? And again, like I said, I'm very kind of biased here to the juice category, right? But if you look at it, even some of these juices end up as a flavoring component in another type of beverage that may not be necessarily considered a juice. So these all kind of come about from the environment. Microorganisms are present, in the harvesting operations, orchards, fields. And so, that's where one of the entry points for these microorganisms is they come in through, the raw material itself. Then when we actually bring it into the processing environment, those microorganisms can actually take up residence in the processing environment. If we don't have, a good cleaning system or anything of that nature to basically deal with these microorganisms, we can have, then the processing environment causing a reintroduction of some of these microorganisms into our finished products, which can become a problem. So really, the types of microorganisms I always like to point out, we have various ones like the good ones, the bad ones, and the ugly ones. And so the good ones would be things, as Omar was kind of talking about. You can have fermented beverages and things of that nature. And then also you can have, things like liquid, yogurts and things of that nature where you can have probiotics in there. So those could be good things. We could have, certain yeasts, if we're talking about alcoholic beverages, that can actually be a good type of microorganism that give us the desired effect. Those are all good ones. We have the bad ones. So those are the pathogens. And I'll talk about those a little bit more in detail here. And then we have the ugly ones, and those are the spoilage organisms, the ones that necessarily don't cause a illness issue, but they can cause, a product quality issue, which can still end up leading to, a recall or something like that in terms of, a quality issue or quality problem that somebody may want to pull that off the shelf and not have it out there. So from a pathogen standpoint, pathogens are microorganisms that cause the foodborne illness by ingesting them directly. You can ingest the cells, and that causes a foodborne infection, or you can ingest the toxins that they create, which is a foodborne intoxication. We're talking about beverages specifically. A lot of times we see E. coli 15787 in the past has been associated with this, especially in juices. So gastrointestinal illnesses and hemolytic uremic syndrome come about from E. coli 15787 infection. Hemolytic uremic syndrome is a very serious illness. It actually causes a shutdown of the kidneys due to the production of Shiga toxins by E. coli 15787 and other Shiga toxin producing E. coli as well. So it can require hospitalization. It can be pretty serious. Salmonella is generally limited to gastrointestinal illness, but can become pretty complicated if you have for instance, a antibiotic resistant salmonella that can cause a problem. So all of these, are foodborne infections where the actual microorganism itself causes, you to become ill. And then we also have intoxication examples. With regard to the beverage industry, we deal with aflatoxins coming from Aspergillus, ochre toxins coming from Aspergillus and Penicillium, and then various different types of molds can produce patchouli, which is another toxin that we deal with. So, as I mentioned, a lot of the processing parameters and things that we have in place and processing technologies that we have to implement for beverage manufacturing, especially for juices, really kind of comes about from outbreaks such as the one that actually kind of helped along with the implementation of juice HACCP. And so if we go back to 1996, we looked at the foodborne illness outbreak associated with E. coli 15787 and Odwalla apple juice. Took the life of one child in a Gymnastad. It sickened 65 additional people and caused hemolytic uremic syndrome, at least 12 more individuals. And so, as a result of this, Odwalla pled guilty to violating federal food safety laws and ended up having to pay $1.5 million for this particular outbreak. So this really kind of drove juice HACCP and really made processing technologies and process validation really a key point for certain parts of the beverage industry. And so I'm going to focus a lot on that here, I know, in my upcoming slides. So again, really kind of focusing on this particular aspect of the beverage industry. So as we know, juice HACCP really came about from, slides get there. Juice HACCP, the initial proposal came out in 1998 after that outbreak. It was that FDA proposed to require that juice processors apply HACCP principles to fruit and vegetable juices and juice products, just as seafood and meat and poultry had done. That was also being implemented around that same time there too. And in 2001, January 2001, we had the juice HACCP final rule that went into place. It required all juices and juice ingredients and beverages to be processed under HACCP as set forth in 21 CFR Part 120. And so a key component of this is the 5-log pathogen reduction performance standard for juice. And I'm going to talk about that when I really talk about process validation coming up during the next few slides. And so as a result of that, all juice processors must meet the 5-log requirement through treatments that are applied directly to the juice. Exception is citrus. Juice processors may opt to use treatments on the fruit surfaces under very specific circumstances. But we help a lot of people in the beverage manufacturing industry to demonstrate that they actually are able to achieve this 5-log pathogen reduction performance standard. And so that's what a lot of our group, when we're dealing with beverages, that's a lot of the work we do and how we help people out in that regard. Spoilage is also a problem, obviously. We have microorganisms that can cause product quality issues, such that it's no longer appealing to consumers. That reduces the shelf life of the product, may result in a recall. Actually recall of your own, I'm not talking about FDA required recalls, but a recall that may result as a problem with regard to the spoilage. And so we've had spoilage organisms in beverage manufacturing, specifically juices that involve lactic acid bacteria, acetic acid bacteria, and Alicyclobacillus. And I'll kind of really highlight that one coming up because that one is also a problem for even thermally processed beverages. We also have fungal spoilage organisms such as yeast. As I mentioned, yeast can be a good organism for us in terms of alcoholic beverages, but it can also cause spoilage problems. And also we have mold as well. In addition to being a pathogen and producing mycotoxins, we can also have mold spoilage issues as well. So some of the spoilage problems that we see in beverages, like I said, from lactic acid bacteria, primarily we see lactic lactobacillus and euconostoc that come through on these, and they can produce off flavors similar to buttermilk. And we also get metabolic products such as lactic acid, formic acid, acetic acid, ethanol, and carbon dioxide, and that can all drastically change the flavor of the beverage as a result of them being present. We also have acetic acid bacteria, primarily Acetobacter pasteurensis and Acetobacter aceti. And those produce acetic acid from ethanol. And so it kind of gives a sauerkraut or buttermilk off flavor and can cause browning of the juice or beverage. And then we have Alicyclobacillus, and this one is particularly problematic because, lactic acid bacteria and acetic acid bacteria are both very susceptible to heat treatment. So you can actually reduce those off very extensively with the pasteurization process. But Alicyclobacillus is actually a spore-forming organism. It's also very tolerant to acids. It's kind of got a double whammy in terms of its capabilities to survive a lot of the things that we're going to throw at it in beverage processing. So it's not controlled with standard pasteurization, and the characteristic spoilage issue that we have with this is glycol production, which basically causes an off flavor. We did a blog post about this, and you can actually go and read more about that and how Alicyclobacillus finds its way into beverages and how you can control it and detect it, and looking at those different things about it in pretty good detail. I have a blog post there that you can go to that link, and that's on our certified group website. From a yeast standpoint, we can actually have Saccharomyces, Candida, and Zygosaccharomyces that can cause various spoilage problems, including carbon dioxide production, alcohol production, that increases turbidity and flocculation of these beverages, and also can cause off odors and changes in color. Again, as I said, controlled yeast growth, allows us to produce wine and beer, but uncontrolled yeast growth can cause all these spoilage problems. And then we also have molds. So the primary molds that cause problems for the beverage industry are heat-resistant molds. They form heat-resistant structures called ascospores. And these can actually survive, a thermal process and then end up in the final product. Over time, these can produce gas. They can form colonies and floating mycelia on the surface. That can change the odor of the juice. They can also produce certain enzymes that are disintegrative and pectinolytic. So things like amylases, cellulases, pectinases, and proteinases. And this can cause, some changes in the viscosity of the beverage and cause things to separate out or change the texture of it. So these are all problematic with regard to the enzymatic activity that they can produce. So with regard to detection technologies, so when we're looking at pathogens, and talking about the pathogens that have caused problems for us in the beverage industry, salmonella and diarrheogenic E. coli, if we look back at these and look at what methods we have, the idea is what we're trying to do here is detect even a single cell that could be present in the sample. And so we have to also remember that because of the beverage manufacturing process and all the different stresses that we're providing to these cells, they're probably going to be in a very stressed or injured state or in a low metabolic state. Heat treatments, acid production, all these different things that we're using to, basically preserve the product can also cause stress on the cells. So if we're needing to actually detect these, we have to keep in mind that we do need to allow for enrichment of those cells so that they can grow to numbers that we can actually then detect with some sort of downstream detection methodology. And so those can be cultural methods or rapid methods. If we actually go with the FDA BAM methods, those are cultural methods. If we're talking about FDA salmonella method, that's going to be a straight cultural method. You can see the enrichment procedure that is used for that there down below. As you can see, it does take us 24 hours to do that pre-enrichment, and then we go to a secondary enrichment with tetrathionate drop to do any type of detection there for a beverage type product. And then for if we're looking at E. coli, we can do a actual PCR method, I can say, from scratch PCR method, but it does take a lot of time to put that together, and it's not like really kind of an out of the box methodology that you can use there. So a lot of times what we have our customers go to is using detection technologies that are rapid validated methods through AOAC. And so those are much faster, and they have been shown to be equivalent performance to FDA BAM methods as a result of being put through the AOAC validation process. So if you look at beverages and juices, we have several different methods that are already validated as rapid methods. So for salmonella, for AOAC validated methods, we have the Hygienovax real-time assay for salmonella. It's validated for orange juice, so we can also use it for other acidified beverages, acidic beverages. And then for biomeria vitis, we have the Salmonella Easy methodology there that's also validated for orange juice. So again, acid beverage products here that we can use these different technologies for. For E. coli 15787, same types of products. We have the Neogen 3M molecular detection assay and the millipore sigma assurance PBS E. coli 15787 assay that are also validated for various beverages, including juices. So we have these methods that we can actually utilize for doing rapid testing. We don't have to necessarily use the FDA BAM methods that are slower. So for spoilage technologies, these are compared to pathogens, fewer and far in between. So really people are not as driven to develop detection methods for spoilage organisms as they are for food safety. So these kit manufacturers really put their R&D efforts more toward food safety. But there are some people actually out there who are looking at doing this now and actually developing some of these more rapid testing methods. If we look at compendial methods, we will see, sorry, it jumped there for me. If we look at compendial methods like FDA BAM and the compendium for the microbiological evaluation of foods, most of those are agar plate-based methods, and so they're slow and they cannot detect lower levels. And so we actually have looked at now using some more of these new technologies like PCR assays, such as the hygiene of back system, the PCR assay for yeast and mold, the hygiene of food-proof beer screening kit, which targets a lot of different yeasts that cause beer spoilage. We'll also look at the Invisible Sentinel Veriflow Decora kit and the Tag Genetics SP spoilage kit. They're all different new technologies that are out there that are focusing on spoilage, particularly in the beverage industry. For Alicyclobacillus, we're also seeing a lot of the development of new kits. It's common to use IFU method 12, which is a cultural method. However, we are getting to see new rapid methods for Alicyclobacillus out there now, such as the Biomeria Invisible Sentinel Veriflow Alicyclobacillus kit and the Hygiene of Food-Proof Real-Time PCR kit for Alicyclobacillus. So this is kind of nice that we don't have to necessarily use the cultural methods for Alicyclobacillus more. You can actually now use PCR methods that will actually do detection and amplification of the DNA that is found in Alicyclobacillus to help us get a faster method result for those. So that's on the detection front, kind of what we're seeing, new technologies, new trends there. Now we'll talk a little bit more about processing technologies that are taking off that we kind of seen that are non-traditional. So traditional control measures, if we look at these, if we're talking about the 5-log reduction performance standard, traditionally we're using pasteurization, thermal pasteurization process with the heat exchanger, doing the LTLT or HTST thermal pasteurization process. But if we look at this, FDA did not mandate that thermal processing is needed to do that 5-log performance standard. You can use other non-thermal technologies. And so that's what I'm going to talk about here on the next slide. Still, thermal pasteurization is very effective in proven at attaining the 5-log reduction in pathogens, but you have the downside on product quality. And as Omar was mentioning, there's a lot more push for minimal processing. People don't want to have products that are cooked to a very, very high temperature. You're looking at things that are kind of gentler, I guess you would say, on the product. But we still have to attain the 5-log reduction performance standard in certain beverage products that fall under the these HACCP regulations. So what kind of non-traditional control measures are we seeing? I would say novel control measures, but I don't want to say novel because some of these have been around for quite a while. So I'll call them more non-traditional. And so, what we're looking at, a lot more so now, we're getting a lot more requests to validate our high pressure processing systems. So we're using non-thermal technology. It uses pulsing of pressures. That's what the high pressure processing is. That causes disruption of bacterial cell envelopes, and it ends up causing lethality to the cells. And then we may also look at using pulsed electric fields. This is using short bursts of high voltage electric fields, and that actually deactivates the microorganisms. Then ultrasound. So ultrasound has a lot of different functions here too, but we're seeing mechanical stress of the ultrasound on cells. And this also, basically because of disruption of the cell membrane, we're also seeing radical formation that results from the ultrasound itself too, that actually also causes stress on the organisms. And so we can actually even combine some of these. I've seen for Alicyclobacillus processing where we actually can combine high pressure processing and ultrasound and actually do very good in terms of getting reductions for Alicyclobacillus, which is not easy to do. So this is kind of actually maybe even better off in the thermal process for something like that because of the way that these technologies work. We also have other non-traditional control measures like high pressure homogenization, ultraviolet light, and pulse light that are also being used. But these first three are the ones that we're primarily seeing the most. So if you want to use one of these non-thermal technologies, you have to think about validation. And this is one of the things that we can actually help with in our group. So you have to scientifically validate the efficacy of your process to show that it's achieving its intended effect. And if we remember the three elements of process validation, we look at process design. It's basically what is the process, what is the product, how do we actually, perform this process, and what are we going to do? Then we have to do the process qualification. That's the part where we can actually help, but we're looking at, actually does the process achieve its intended effect? That may require us to actually do a process validation study. I'll talk about that. And then we have to do continued process verification, ensuring that the process is actually still continuing to do what it's doing, that we haven't changed any parameters or caused anything that would adversely affect how we are achieving our intended food safety objective here. So the question becomes, do you need process validation for a beverage product like this? So if we're talking about some things like thermal pasteurization, those are considered safe harbors, and so you don't necessarily have to do a validation for that. If you can do a verification but not a full validation, and you can get a process authority to review supporting data, as Omar was hinting at earlier with regard to commands and how you use those and making sure you have a process authority review on that is good. You definitely need to have that happen, but you may not have to do a full validation for something like that. If it has a thermal pasteurization process that's considered a safe harbor. If we have, insufficiently publicly available data and or regulatory guidelines, such as these non-thermal processes that are non-traditional, you may need to generate some data, and you need to have a process validation study on file to be able to justify your process and how it's working. The problem is how do we perform one of these process validations? The conundrum is we want to use pathogens for this type of stuff. We can't introduce those into a facility. So the solution is really to kind of either come up with a laboratory simulation of the process with the pathogens, and that can be very difficult to do. Especially if we're talking about something like a high pressure process. We don't necessarily have one of those pieces of equipment laying around the lab. And so that can be very difficult to simulate a process. Now, if we're talking about, kind of maybe a thermal process or something along those lines, it's a lot easier to simulate that in the laboratory with time and temperature. But if we're doing like high pressure or pulsed electric fields, we may actually have to do that in the facility. And so in those cases, we would have to validate a non-surrogate or non-pathogenic surrogate organism to go in and take and use in the facility that would act the same way as the pathogen. Or, the option here is to also perform a pathogen inoculation in the pilot plant. We have to be very careful about that because that could still cause problems, especially if, that pilot plant is going to be used for R&D purposes and somebody may be consuming that product. We would have to make sure that that's properly decommissioned after we're using the pathogen in that particular facility. So if we're going to do a validation, how would we go about doing the process? If we're doing a process validation study for somebody, we're going to, obviously visit with them and, find out about how this is actually going to be going about, what kind of process they're doing, where's the best place to do it, as I mentioned before. But once we get past all of that, we're going to kind of follow this general process for doing the validation. So we're going to inoculate the product with a pathogen or a surrogate organism, depending on where we're actually doing the validation. We package that product if applicable, like if we're going to go to an HPP process where it's going to be treated inside the package, we definitely have it in the package, in the final product package. Then we're going to run through that process. We have to keep in mind key considerations of temperatures, dwell times, batch sizes, internal temperature and product attributes, come up and come down times. And then we're going to return those samples to the laboratory as quickly as possible, keep those chilled. During that process, then we're going to perform a quantitative analysis method on these. So it's generally some kind of a plating procedure or an MPM procedure to be able to see what sort of reduction we were able to achieve with that process. And so if we're validating these novel, non-traditional processes, we're going to go through this and actually see, does it actually meet that 5 log reduction based on this validation study that we performed. So hopefully this gives you a little bit of an idea, about what needs to be done if you're wanting to implement one of these new processes and how you can actually use it. So future perspectives here, as Omar was mentioning, we have a lot of new trends in the beverage industry. We have new desires from consumers, and they have product lines that continue to expand. And so those beverage processors are going to continue to face different challenges in controlling microbial pathogens, these spoilage organisms. And so we're going to have these detection technologies continue to develop, especially on the spoilage front, and also pathogen technologies that are faster and easier to do larger volumes. That's one thing we're also seeing here now too, is when we test larger volumes of these products. And so, as these new technologies for control are gaining hold, we definitely need to keep in mind about how we need to validate these technologies so they can be adopted and be able to show that we're meeting the performance standard requirements as needed. So I think that's it. We have, I think, a few minutes left for questions, so I'll go ahead and stop sharing our screen. We'll go ahead and open it up, I think, for some questions.
Thank you, Alex. I think I can go through some of the questions. I kind of place them here. I'm going to try to cover. We don't have that many questions, but the first one is, if not covered, can you talk a bit about PFAS? Some customers are asking about requirement for PFAS apparently in juices. So PFAS, the polyfluoroalkyl substances, per and polyfluoroalkyl. These are really complicated substances because they have a fluorine bond ion with molecule with a carbon and create a very, very steady bond that is really resistant in the environment. So FDA is doing a lot about PFAS, a lot about testing. They have contract with different states. So we are kind of at the early stages of what is even the ideal testing for PFAS. So it's kind of hard to really navigate this and say, oh, this is what we need to test and this is the amount or something like that. We don't really know that. You have to understand that FDA is trying to understand PFAS in food, but PFAS are using many other things. Like, for instance, people that are making a sport garment for sport, water resistant, there may be quite a bit of PFAS there, okay? And FDA has certain approvals for it. So kind of complex, there is not a single answer, there is not a straightforward answer yet. Can you talk about chlorates testing? No, well, chlorates is very well recognized. We know very well when chlorates can be. It's an oxidizing product that in water you may find it because of the type of water treatment that you use. If you use chlorine dioxide or if you're using a calcium hypochlorite, for instance, you may have that as a byproduct, but it will depend also on the ions that you have in the water. So whether the water is hard or soft, there is a lot of testing. We're not going to go into that because it's very specific. There are questions about whether they can get recording of the webinar. Yes, I think there will be, the recording will be available, correct, on the internet, Jessica?
Yes, I believe we will have it available on demand.
Perfect. Then there is one about pharmaceutical companies asking for a specific international. So we are not covering pharmaceutical products here. We're talking about food, okay? So that's a different, they have a different type of regulation and different type of stringency. This question is for you, Alex. How is a test method validated and by whom?
Yeah, so test methods are validated by comparing to a reference method. So when we do these method validations, they're overseen by AOAC. Generally, it could be Health Canada, could also be here in Europe. They have their groups there as well. But basically, we compare a rapid methodology to a reference methodology by taking samples that are inoculated at the same level and then doing testing with both platforms and doing a statistical analysis to show that they're similar enough.
Great. So because we are just running out of time, I'm just going to entertain probably one or two other question that may be applicable. We do provide processing authority letter. There's one that says, how long is the process for a processing authority? We're kind of fast, but you know, we can talk about that later. You can reach us out. It depends also on what you're looking for. Remember, the processing authority, the term comes from a specific regulation, 21 CFR 113, that is low acid canned food. And then we have been borrowing that and kind of overstretching the concept and sometimes distorting it. So it's important to understand that if there are many different ways to create a processing authority letter that conveys what a co-packer may need or what the a client may need or a supplier may need. Okay, I don't see any other a specific question. I don't know if you want to add anything else, Alex.
I think we're good. You know, I think if you guys have any questions, you can have our contact information. You can go ahead and get in touch with us after the webinar. We'd be happy to answer any questions. And you know, again, if you need any sort of services, EAS Consulting Group can help you out on the consulting side of things, and here at Certified Group in the laboratory side of things, we help out with process validations during the lab legwork. So we work a lot with Omar and his team from the consulting side and doing validations for folks. So if you guys need help in any kind of regard, we can definitely help you guys out. Just want to make sure you guys are aware of that.
Well said. Thank you very much. Thank you. It was great sharing the time with you, Alex, also.
Thanks, Omar.
Thank you, everybody. And as mentioned, we will have a recording of this available publicly on demand in the coming days. Thank you again so much for joining us today, and have a great rest of your day.